All right, today we're going to talk about two things. I'm starting over again because I'm going to record. Two things, nerve impulses and how neurotransmitters allow nerves to communicate with each other. First of all, this word self-propagating. A self-propagating wave is like when you touch a domino, like I said just a minute ago, and you touch that first domino and then the dominoes keep falling. They keep themselves going without you having to keep touching the dominoes. This is exactly how a nerve impulse travels down an axon. And so let's do a little drawing really quickly. Let's draw a... It says the network is down. What? Okay. Still recording here. So we've got this line here is the nerve cell membrane, right? This is the axon membrane. I'll draw the other side. Okay. And across that axon membrane, you have the normal conditions are that you have a positive charge on the outside and a negative charge on the inside. So this represents our before. This is our resting potential. We call this membrane a polarized membrane, okay? Because there's a positive side and a negative side. And this is the inside of the axon. And so then you can obviously tell that the outside of the axon is on the other side of the line. So does anybody remember from your reading what happens to depolarize that membrane? Okay, because some stimulus, it could be a mechanical stimulus or a receptor mediated, like a nerve channel opens up and allows ions in. Sodium ions with a positive charge begin to flow into the cell. If you have a flood of positive charges going inside of the cell, what's going to happen to the conditions inside of the cell? It's going to become positive. Okay? So when the nerve depolarizes, we no longer have that natural plus outside and negative inside. We have a rush of positive inside, which makes the outside slightly negative compared to the inside. And that is the action potential. That wave of depolarization travels from the cell body end, and remember we also call that the soma, travels down the axon towards the axon terminals. Okay, so it goes from the cell body end with the dendrites to the axon terminals where the axon ends and it branches out and attaches, or doesn't attach, but comes into close contact with other axons, the axon terminals. So here's this word, depolarize. So we have a polarized membrane at our resting potential. When the sodium floods in, it makes the inside of the membrane more positive than the outside, and so it depolarizes. And then lastly, in order to restore the membrane to its resting potential, potassium ions, which also have a positive charge, flow back out, making the outside again more positive compared to the inside. And this is called repolarizing. So we have at rest a polarized membrane. When the action potential slides down, You have an area that will become depolarized, and then in order to restore, you're going to pump that potassium back out, which will repolarize. Now, in order to do this, this is an active transport process. Because you're pushing ions against the concentration gradient. That sodium is actually being pushed against the way that it would naturally want to go. So it requires energy, lots of energy, in the form of ATP. Do you guys remember which organelle is responsible for the mitochondria? So nerve cells are going to have lots of mitochondria because they have high energy needs. That means they're going to require lots of glucose and oxygen. So that's why we need to breathe. That's why we need to eat. The nice thing about watching the video is that you can always pause it and draw. So now we're going to look at this back in words. So self-propagating, it keeps itself going. It's an electrical disturbance. It's really an electrochemical disturbance because it's not electrons flowing. It's a change in charge caused by the movement of ions. So it's chemistry creating an electrical current. In words, some stimulus, maybe it could be, if it's a touch receptor, it could be mechanical being stimulated that way by the membrane being bent. Opens those sodium ion channels. The sodium moves in, creating a slightly negative exterior and a positive interior. Okay. So as I drew on the last slide, you have a negative on the outside, a positive on the inside. And so this picture is a neater version of what I just drew, right? So what would the yellow arrow represent there? Okay, the sodium being pushed in. Is this, let me ask you this. I'll say at position 1, at position 2, at position 3. At position 1, would we say that that is a repolarized membrane, a depolarized membrane, or resting potential? Resting potential, good. Two, depolarized, repolarized, resting potential. This is repolarized. And then that leaves part 3, this part 3 right here. This is an area that is being depolarized. Axoplasm, that just represents the plasma inside of the axon. And this is just showing the net movement of the sodium and the potassium. Okay? So if you're at home, you are going to watch the video now. For everyone else, we're going to watch the video, but I'm going to pause the recording. Okay, so hopefully those of you who are watching at home stopped the video, listened to the animation and took the little practice test and reinforced those concepts. Okay, so here we're looking at two neurons. The one on the left has insulation on its axon. We call that the myelin sheath. And the one on the right is unmyelinated. There it points to the red cells that form the myelin sheath, and it refers to them as Schwann cells. So is that axon in the central nervous system or the peripheral nervous system? Peripheral. Why would you say peripheral? Why? Because that cell is called a Schwann cell, and we only have Schwann cells in the peripheral nervous system. See, I've been studying Ms. Zitterkoff, and I know that the cells that form the myelin sheath in the central nervous system are called oligodendrocytes and not Schwann cells. So I know, therefore, that must be a neuron that's in the peripheral nervous system. See, that's stuff that I would put on your quiz tomorrow. Remember the glial cells?
Yeah.
I know that was way back last week. I know. So we have different cells, myelinated versus unmyelinated. Which ones make up white matter in your brain? Myelinated axons. And gray matter is unmyelinated axons. And so, what's the difference between how an action potential runs down an axon when it's myelinated versus when it's not? When you have a myelinated axon and the action potential runs down the axon, yes, the sodium ions go into the membrane at the space in between called the node of Ranvier. If we go back here, oops, too far. Do you see the node of Ranvier pointed out on that diagram? That's the space of the axon in between the myelin cells. Okay? When you have an action potential going down those types of axons, the sodium only rushes in at the spaces where it's not wrapped or insulated by those myelin cells. And so the sodium rushes in there, and it pushes the potassium out of the way. Because remember, like charges repel each other, right? Opposites attract. So the two positive ions, the sodium comes in and it pushes the potassium out of the way. The potassium then moves through the inside of the cell down the axon and causes the axon to depolarize on down farther, which leads to a much faster action potential going down the membrane. Because it jumps from one node to the next, this is called saltatory conduction. This word is not in your notes, I don't think, but it needs to be. I think your book does something kind of sneaky because this word is not obvious that you should understand it, but then it appears in the review question at the end of the chapter. It's in bold on page 174 of the book that we have in class. It is in the section. And it is quite significant because saltatory conduction is what allows nerves that are myelinated to transfer their impulses much, much faster. This also contributes to the significance of a disease like multiple sclerosis, where white blood cells attack and destroy the myelin. And
Wait, what does it allow nerves that are myelinated to what?
they can transmit their impulses much faster because the potential literally jumps from one node to the next. In a myelinated neuron, look at this picture here. In a myelinated neuron, the action potential can jump from one node to the next. Instead of like in a non-myelinated neuron, it has to travel like that, dominoes falling. Yes?
What do they transmit? I'm sorry.
What do nerves transmit?
Oh, duh. Okay, sorry.
So a myelinated axon can transmit a nerve impulse much more quickly because of saltatory conduction, that the impulse jumps from one node to the next. But in a non-myelinated axon, it has to travel all the way down the axon like dominoes falling over in a row. Here's another picture that shows where the sodium inflows, and then the action potential actually travels through the axon, not across the surface of the membrane. So it jumps. So let's see, I'm going to pause the video, make sure that you can answer these questions. I want you guys to stop and talk about them real quick, and then we'll discuss them. So the next part of our lecture is about neurotransmitters, which is the end result of this action potential traveling down the axon. What is a neurotransmitter? It's a chemical compound That's great. It's released by the axon terminals. It's held up in vesicles of these presynaptic neurons. So we're going to see a couple of words here, presynaptic and postsynaptic. Pre means before, post means after. So the synapse is perhaps the thing that you forgot. So here is neuron A. Okay, this is the soma. This is the axon, and those are the axon terminals, right? And then here I have another this is my soma, my axon terminals, right? This space here, because the nerves don't touch, they get really, really close. That space is called the synapse. So that space there. So this would be the presynaptic neuron. And this would be the postsynaptic neuron. See? Presynaptic neuron, postsynaptic neuron. So it's across that space that these chemicals are released, and the whole point of the action potential traveling down is that when it gets to the axon terminals, they're stimulated to release those chemicals. So these neurotransmitters bind on the surface of the postsynaptic neuron and open ion channels, which then, guess what kind of ion channels? Sodium ion channels, which allow sodium to rush in and create a nerve impulse. So serotonin is one of the common neurotransmitters. And that neurotransmitter is what is responsible for feelings, emotions, hunger, depression, if you have low serotonin levels. And so you guys have heard of things like Prozac, right? Prozac is an SRI, serotonin reuptake inhibitor. SRI. It's a category of antidepressant. So serotonin, you just learned, is the name of a specific neurotransmitter that's involved in happy feelings. Reuptake means that after the presynaptic neuron releases its neurotransmitter, it reabsorbs it and brings it back in. That's called reuptake. Okay? So the presynaptic neuron takes the neurotransmitter back in. And to inhibit means to keep something from happening, right? To stop it, to impede it. So if you're stopping reuptake, then where is that serotonin going to be? So here's your synaptic knob, and you have these vesicles that have this serotonin in them, and they move to the edge of the vesicle, and they fuse with the vesicle, or they fuse with the membrane, and they release the serotonin out into the space. And this is your presynaptic neuron, and this is your postsynaptic neuron. Didn't you draw this? This was what you were supposed to draw on your second page of your web quest. And so this neurotransmitter, the serotonin, would then float across the synapse and bond with a channel, a receptor, in the membrane of the postsynaptic neuron, which would open a channel and allow sodium to flow in, which would then cause it to send off its action potential. Well, we have a serotonin reuptake inhibitor. Now, this serotonin is not going to be taken back in. So where's it going to go? It's going to stay in the synapse. If it's in the synapse, what's it going to do? It's going to keep stimulating that postsynaptic neuron. And that postsynaptic neuron is going to keep firing signals that indicate well-being, that everything's okay. Do you see? If you don't have enough serotonin, you're not getting those signals of happiness, of wellness. And so on a chemical level, that's why depression occurs. You can't just take yourself out of it. Chemically, you're out of balance in your brain. And now I'm not talking about you just have a day where you feel kind of down in the dumps. I'm talking about serious clinical depression and anxiety, where you just can't get yourself out of it. It starts to affect your life. Okay? And it comes out different in different people. For me, I would hold it all together at work, but at home, I would lose it with my kids. You know? Because I couldn't lose it here, even though I had feelings of, oh my God, I don't know how I can do this, you know. And these medications helped me feel calmer than I've ever felt in my entire life. I didn't know what it was like to feel like I can handle this. Like, you know, I would have horrible thoughts. It's called catastrophizing. And I would freak out about things that I shouldn't freak out about and make myself really unhappy. And it's not healthy for you too, right? So I'm going to show you this link here goes to a crash course video. We'll go back to it in a second. So one of the questions I want to ask you is, so when you release the neurotransmitter and it binds and you get a cellular response, how does that cellular response stop? Reuptake, so it can be taken back in, okay? That's shown by 3A. Degradation means it degrades, it breaks down. Or C, diffusion, it just diffuses and spreads out and it's not there in enough volume to stimulate the postsynaptic neuron anymore. I didn't have this slide on the presentation for my other class. I just added it. Okay. So here's your questions. I want you guys, we're running out of time in class. So it's more important right now that we go through and we look at these questions. Tomorrow I'll try to remember to show you the video or watch it at home. It's 10 minutes long. Okay?